Welcome everyone to this autumn's round of innovation talks. I think we've had quite a bit of a summer holiday since we did the last. These innovation talks is really where we enjoy inviting innovative initiatives, consortia, researchers, regulatory, governmental agencies to present their ongoing work that within this frame is related to health data science as a very, very broad term. The purpose is really here to raise awareness about initiatives, either they're local or global, related to life science broadly. The common denominator should really be health data and advanced analytics as part of the topic to be featured here. We really hope that the presentations that we share here and also those who are accepting to come and present will help to foster interest and a knowledge sharing about health data science innovation. And as we're doing this, not only in the greater Copenhagen area where we are placed, but really to stimulate a global interest and ability for initiatives to reach out to each other. So it goes broadly for those of you listening from the outside, watching this. Feel free to reach out to the email address here, which is for Lars, who is hosting the meeting right now, to be featured in this series. We can offer you that platform and invite you in to showcase your initiative. Just a disclaimer here that the presentations are not endorsed by the Danish Medicines Agency, and it will actually neither be able to solely present a commercial offering, so it's not a commercial platform. With us here today, we have the privilege to welcome Exscalate, which is a European Commission Horizon 2020 initiative. Which is a public-private consortium of really looking at how to apply supercomputer resources, artificial intelligence to explore and research new treatments and drugs, and especially with Exscalate4CoV. So obviously we are now still in the pandemic, although many people have been vaccinated, but there's still a massive need to understand further treatments and explore good treatment options for those who then, despite of infections and vaccines, do end up in a situation where they require care. But I think I would just really love to leave it to the good Exscalate team to kick us off on the innovation talk today. So over to you.
Thank you very much. First of all, for the kind invitation. It's a privilege for me and for my colleague Carmine to be here and have the opportunity to present our platform and our results from the Exscalate4CoV project. I'm trying to share my slides. I hope. Can you see the slides?
Yes.
Perfect. Also in presentation mode. Okay, so before entering in the presentation is organized in actually in 4 parts. So the first part is an overview of the status of the pharmaceutical research in terms of outcome and the capacity to address medical needs. The second part is dedicated to the platform and also with a live demonstration of the platform in action through virtual reality, thanks to Carmine Tallarico. So a slide that want to represent the direction, the evolution of medicine as we know it at the moment is from modern traditional medicine. Now we are almost, and is a question here if we are really in the era of precision medicine with the final goal to arrive to predictive and even preventive medicine is where we are able to address the target population and eventually take action before that some kind of pathologies do occur. I have 2 slides where after saying that only the less than 10% of a molecule do arrive on the market and the patients, I want to stress on the concept what happens when you have a successful molecule that survived to the clinics and arrived to the patients, with two examples. The first one is well-known breakthrough disease-modifying agents, Enbrel, that is really changed the behavior of the pathology. But if we observe the response of the population, we can see that only the 23% of the population have the 100% of recover due to the administration of Enbrel. Then we have the 60% of the population that have less or more than 50% response. And then we have 10% of the population that do not any effect from the Enbrel, and the 7% of the population shows a severe side effect that to stop the administration on the drug. And for me is quite a good example talking of a biologics that is a very precise effector of the system that maybe in this case we are in the presence of multiple different kind of rheumatoid arthritis that we call rheumatoid arthritis an ensemble of different mechanism that in one case is completely controlled by tumor necrosis factor that is the fully responding population, then other effect come into play. So the central dogma of the pharmaceutical industry is one disease with the selection of one target that produce one single drug to address the problem, but in many cases some important limitations. So the understanding and the modeling of the disease is fundamental for identifying the responding population. Another indication that I find very interesting that move to the patients and diversity and complexity and peculiarity of the patients is the Huntington disease. Huntington disease is a monogenic disease. So in this case, we have a single mutation on a specific protein, but this produces a pathology that is multifactorial. Also in this case, I want to stress on the point that if you see in the plot the severity of the mutation on the axis and the onset of the pathology, you can see if we fix on, for example, 40 mutation, you have a very broad onset of the pathology from 30 years to 70 years. And from a pathology that after 10 years from the onset arrive to the death of the patients is very important if you have the onset at the age of 30 years or the age of 70 years. So there are many conditions from a pathology that environmental and genetic the onset that do affect this. And it's important to take into account when we try to address this population, because probably it's possible to act on other effect than the protein itself that is mutated to push the onset to an earlier age instead of try just to address the single mutated gene. And then we arrive to this slide that is reported in Nature not so many years ago. And what is expressed here is the number to treat. That is the number of patients that have the maximum benefit respect to the treated population. And you can see then we have still Enbrel in this slide that almost 25% of the population is fully responding. But you see in this slide, here is represented the top 10 selling drugs in the year where this Nature paper was published, but it accounts for the problem of responding population. As you can see, the red bodies are the majority in all the cases. So reaching a number to treat higher than 30 to 50% is very, very difficult. Because actually today what we do is that we do diagnose a disease to an unlabeled, unannotated population, and then we enter in the situation that we have to select one treatment or the other, and the only chance that we have if the treatment A do not respond on the population that is administered, we can to the treatment B. And if it's not successful, even in this case, we have what we call the still unmet medical need. So not responding and person to expose side effect to stop the treatment. And actually is what we call error and trial in this case. And I reported this nice definition for the Oxford Dictionary in what it means error and trial. The outlook for the future, we think that should be something in where we decide together to profile population and patients, and then to try to use the information and the profile of the population to stratify and model disease that allow us to identify responding population for the specific treatment before the treatment, not just after the treatment. In this case, will be of course the best possible solution where the identified treatment are able to solve and remove the medical needs for the specific pathology. In this case, we had to move to something that is on purpose designed medicine, but it doesn't exist. And I think that altogether, pharmaceutical company and decision makers had to understand how to arrive to this scenario where it's possible to address in a more systematic way the disease and the patients. And of course, to do this, we have to act at a politically and regulatory level. We have to change the way we do clinical trials and also preclinical activities, starting from drug design and eventually repurposing in a way to maximize the level of information and the translational of the models from the in vitro to the in vivo to the clinical trials, using at maximum the open data approach from clinical trial, but also for other kind of data that are fundamental for arrive to the goal of predict the responding population and develop effective treatment. We just focus in our presentation to the smallest part and the less complicated part of the story, that is the design of new drugs. But I want to stress here the concept that the tools that we use for novel drugs can be used also for repurposing. And we really believe that repurposing and reapplying molecules that have already been used and exposed to the population is very important to address neglected and rare disease, for example, but also to limit the cost of the treatment in the case where possible to reuse molecules that have already been identified and the cost have already been addressed by measure indication in the field. So our mission is to try to improve clinical trial success through the implementation of polypharmacological profile using the highest quality chemical probes, and all these supported by artificial intelligence and molecular simulations. The Exscalate platform is composed by three major parts. One is related to the chemical libraries. To the other side, we have the targets. And in between, we have the simulation engines that is able to take one molecule and evaluate its possibility to interact with a target in a very efficient and effective. Now I pass the floor to Carmine that show you in a live demo. It's possible to combine all these things in our day-by-day work. So Carmine, please.
Thank you so much, Andrea. Can you hear me? Okay, so I will put on my headset and I will share my virtual room. Okay. Can you see this blue protein?
Yes.
Okay, nice. So as Andrea already mentioned, we had in our Exscalate platform, we had three main pillars. There are the comprehensive therapeutic target database, the tangible chemical space, and our docking software that is Ligand. So with respect to the comprehensive therapeutic target database, we had a very huge number of annotated 3D protein. And in that case, the annotation are related and coming from a knowledge-based part. So in that case, the information are retrieved by using a public database, or the annotation can come from in silico simulation. And for example, we are the possibility to analyze the protein by studying, for example, which are the most important functional domains. We are able to individuate and map which are the cavity of the proteins that can act as binding site, and which are the region of the protein that can be useful for the protein in, for example, protein-protein interaction. And this information that comes from molecular simulation can allow us to study at atomic level, which are, for example, the differences among an orthosteric binding site with respect to the, for example, an allosteric binding site, because we are able, by using in silico modeling, to evaluate the chemical and physical characteristics of a specific binding site in that case. And in particular, we can study at atomic level which are the atoms of the ligands that can interact with the atom composing the protein. So this is one of the aspects that we can analyze by using the information coming from the CTTD. But another very important step is the possibility to analyze a protein in terms of its dynamic behavior, because as you know, of course, the protein is a dynamic stuff that can move to bind a specific protein, to reach specific conformation. So by using a technique that we call molecular dynamic simulation, we can analyze in depth the structural variation in the protein structure that can affect the functionality of the protein itself. And in that case, we are also able to, for example, extract from this, let me say, this sort of video, specific conformations that we can use in our virtual screening campaign, for example. So by combining the information that come from the comprehensive therapeutic target database, that are all information related to the targets and its 3D structures, we can combine the protein, the targets of the comprehensive therapeutic target database with huge amounts of the ligand that we can find in our tangible chemical space database. So in that specific case, we had in our virtual library an impressive number of molecules. We are reaching billions of virtual molecules that can be easily synthesized. And by combining the ligands and the protein, we can act and evaluate a huge amount of ligands in a huge number of targets, in that case of therapeutic targets. So we can do that thanks to our docking, structure-based docking engine that is called Ligand, that allow us to evaluate the virtual activity of our molecule, but we can do that in an impressive speed and quality of the results. In particular, we can screen billions of molecules in a few hours, and this is due to the fact that our docking code is able to virtually screen 4,000 of molecules per second, and we had the possibility to use this software on a high performance center, so we can go through supercomputing center to enable this very huge performance. Then I will conclude with a very interesting information with respect to our capability to use all these stuffs to study, for example, the SARS-CoV-2 and all the molecular mechanisms that occur in during this pandemic, and that we use to analyze and found new therapies. For example, we had here the ACE2 protein into a membrane, and by using these techniques, we are able to analyze the interaction that occur among the ACE2 protein and the spike protein, and in particular, these techniques allow us to analyze which are, for example, the differences that can occur in some specific variants and that can alter or the affinity among the spike protein with respect to the ACE2. So we can now we can study in depth which are the differences among the different spike variants and how this protein can interact with these effectors. So that's all from my side.
Thank you, Carmine.
I will stop sharing. Thank you, Andrea.
Can you see my screen again? I hope you can. So this is a schematic representation of what Carmine said. We have a integrated platform that combines target, molecule, and an engine for evaluate the combination of putative ligands against target. I want not to enter too much in the description of the tool to have the maximum benefit from the Q&A discussion. There is one single thing that I want to take care talking of chemical libraries. So especially in these pandemics, it became evident that the variability of compound for experimental testing is really a limiting step. Because at the moment, as you can see, we have, in theory, more than 10,000 molecules that in the history of medicine enter in clinical or are on the market. And then we have other molecules that are administered to the population, for example, from the traditional Chinese medicine that account for 40,000 molecules. And then we have other molecules that people are daily exposed that are food and nutraceuticals. The point is that really a small part of these are available, for example, for evaluating their ability to interfere with the COVID. Worldwide, only less than 40,000 molecules drugs have been evaluated out of 10,000, just because they are not available for screening. So one point, especially in the future and the preparedness for future pandemics, is to understand that a molecule that demonstrated is non-toxicity or in human is a sort of treasure of the mankind, because we can arrive in a point that we need all that we have developed so far to find in the fastest possible way a cure for a pandemic. And then we have other higher quality libraries that are natural products, oligonucleotides, and then we move to screening compounds, synthetic compounds generated by company, and de novo molecule generated by computer that can be synthesized. For all these chemical space, we collect all the knowledge-based system, as Carmine have already mentioned, about the reference in literature. We tag these compounds about side effect, activity on target, their relation with disease, and of course, all the biological activity that not been identified. Especially for the safe in men subset, we also collect, we are in a way of completing the full profile of the activity of these compounds against all the comprehensive therapeutic database, and then we arrive to more than 1 billion of predicted activities. And at the same time, we use the same ligand and structure-based models to predict cardiotoxicity and neurotoxicity and metabolism for these compounds to have the maximum benefit from computer simulation and modeling. Another part of the story, as already mentioned, is our database that collect the targets. There are many different kinds of targets. Also, in this case, we map all the information that are already available, especially pathway, motifs, domains, and protein-protein interaction. I will come back on this specifically for the COVID. All this information we found useful in finding novel and unreported effect of the spike. And to the other side, we put a lot of effort, thanks also to the possibility to use the European infrastructure or high-performance computing, is to generate massive molecular dynamic simulations using simulation for mapping the binding site and characterize and hypothesize and predict protein-protein interaction site based on protein-protein interaction map generated experimentally. Now, in the last part of this talk, I want to focus on the project that in which we have used all the tools that I have described so far in try to solve the problem or at least mitigate the problem of the COVID. And is the case of Exscalate4CoV project that account for 18 partners and more than 200 researchers involved for 18 months from nicely spread in Europe. And then during the project, we collect other 30 partners worldwide from China, South Korea, United States, and Africa, and so on. And the approach in this case is the same we are trying to use in the last 5 or 6 years, is to address the problem from a polypharmacological point of view. So instead of focusing one single protein, like other companies have approached the problem, also research institution focus, for example, on MPRO, on proteases or spike, we decide to model all the non-structural proteins and the spike proteins. The human proteins are fundamental for the interaction, and as Carmine showed you in the virtual room, also the complex between the spike and the ACE2 in membrane. So we try to evaluate the possibility to a drugs or the nutraceuticals or natural product to be an effector and eventually a multiple effector of the entire system that involve the infection of the COVID, not just focusing on a single event. And after these simulations that identify a pool of molecules that have a suitable in silico polypharmacology, we enter in the validation in multiple assay. And in this case, we are able to identify and prioritize the raloxifene. That is a molecule that also shows direct antiviral activity against COVID in our test, especially the ability to stop the viral replication in a phenotypic assay, but at the same time use the entire knowledge base collected from this molecule that is on the market from the 70s, and the original mechanism of action that is a SERM, so is a selective estrogen receptor modulator, and their effect on the host, and even more relevant, the perfect biodistribution to accumulate the product in the lungs, that is the target organ for the virus. The combination of this information allow us to select and then enter in a clinical trial with the raloxifene. And what we can say that from the design identification to the end of the clinical trial in less than 10 months, we are able to arrive to a proof of concept of the activity of this compound in a clinical trial setting. The analysis of this have been submitted, will be submitted this week to a journal to make all the results available for the community. But what I can say is the data are very encouraging in saying that raloxifene could be an effective treatment for COVID. Other possible application of the platform, and this is my very last slide, is in repurposing in other indications. Also, rare and neglected disease are privileged application for this platform, and the reuse of molecule to the mechanistic identification of novel activities is one of our major activity at the moment. We have submitted with other very important European partners to an Horizon 2020 call that is for the implementation of the European Drug Repurposing Hub. And we also think that we have a lot of possibility to apply these techniques, for example, to personalized medicine. In this case, we are in another European project account for glioma for children. In this case, we model the mutants that are specific to the patients and try to understand if the current treatment are effective or not, also against this mutation. The platform can be used for the reduction of animal testing to the fact that it's possible to derive predictive model mechanistic, so based on targets and not on molecules, and to address safety and liability, and also for side effect prediction, because the effect of drugs seen in their polypharmacological action to the entire set of human protein can address and predict possible side effect. There is a specific application. We are by the end of this month, we will submit a paper that we demonstrate that using our computer simulations and annotation, we are able to identify a mechanism of the spike protein interacting with the host protein that is responsible for one side effect that we know is devoted to the spike interfering with the host. So this is my last slide, and I hope we have a very interesting Q&A discussion. I stop sharing my screen in a second.
Well, thank you, Andrea. Thank you, Carmine, for a very impressive presentation. Large numbers all the way through. Maybe I can kick us off while people think about a question, or just raise your hand if you got one ready already. I think here's Tara, brilliant. Just a quick one I wanted to start out with. So really interesting with that estrogen kind of drug, Ralofexin, Fexofin. Did you try to investigate some of those drugs that through other means kind of became known to have an effect or assumed to have an effect during COVID-19? I think we've had a number of drugs. Did you go back and investigate some of those in your modeling, whether you would have identified those? Would that have been something you would have looked at?
Yes, it is the first outcome. We have other molecules that are under investigation at the moment, and some of these are already used by a large set of the population. So, for example, a retrospective clinical trials is ongoing for 3 of these molecules, because if our assumption is correct, we have to see some kind of protection in the population is already treated. So for Ralofexin, we are not able at that time to have access to this data, but now expanding our network, we have the possibility to have these retrospective clinical trials are ongoing at this moment. We have another molecule very, very interesting that have a wide use in the population, and we believe that if the retrospective study support, we can enter in a real clinical trial for a different molecule, a completely different mechanism of action.
Good, thank you. Tara.
Thank you for the talk, very interesting. So I'm just curious if you also have some machine learning initiatives on kind of projecting or predicting mechanisms of protein interactions. So if you have, I read that you can upload protein structures in the drug box, or you can submit compounds and then see how they fit. Could you also develop functions that suggest possible compounds, or is that also part of it? Because there was a huge buzz with AlphaFold, and if you can kind of combine some ideas, then maybe that's
Yes, we have already developed and will be launched, I think, by the end of next month, a profiler. So all the target that you see in my presentation will be completely exposed to the community for free, obviously. So you can submit your molecule, have the entire profile available for your set of molecule of interest. And in the future, the plan is to make the platform available also for the academic community in a broader approach. So the idea of the company is that once we obtain European funding, so from taxpayer, we have to go back to the community. So the platform is meant to have for-profit activities. That is our internal research and also for our partner. But we plan to have in another dedicated project, European, that is Ligate, that at the end of this Ligate project, we will have a cloud platform available for the research community for free. And in that case, you can select your target, your drugs of interest, and you can use within the cloud platform. Is already part of the drug repurposing hub. In the case will be financed. So we already plan to accelerate the release of this platform from the research community in case of this additional grant from the community. But the idea is anyway to arrive to make it available for research. Of course, there is a topic of CPU time, but we are working with the PRACE and that is a European consortium to try to find a grant in terms of GPU hours to guarantee the researcher to have access at no cost to the infrastructure, because doing this simulation is quite expensive in terms of GPU time, as you can imagine, but can be addressed within the resources available from the European Commission in the HPC area.
Is this your most important, most critical limiting factor in advancing? Is that access to sufficient CPU time?
But for sure, the quality of the simulation is related with the level of infrastructure that you have. Of course, it's more relevant in pandemic scenario. In this case, we also take advantage of other research centers, also private. For example, the Italian oil company give us access to the computer center just to increase the amount of computer power to speed up the process. In situations that are less critical, of course, it's not so dramatically important to have millions of GPU hours. If you have one single molecule with a small number of target, we can do the profile in seconds, also on a PC. If you want to do an extensive profile in terms of 10,000 targets in multiple conformation on 10,000 molecules, then you need an HPC center.
Mm-hmm. I'm just wondering, Andrea and Carmine here, that there must obviously be a interesting overlap with some of the needs in the pharmaceutical industry and what you do here. You started out by showing some of the graphs that I've a few times have seen referred to as the opposite of Moore's law, called Eroom's law, meaning that we see a tendency in industry towards a return investment of zero and longer time to develop new drugs of effect and relevance. How big a potential do you see in this for industry to identify new drugs, but also new targeted populations? I think that's part of your Horizon idea, as I understand the title. Wouldn't that be a funding mechanism for you guys? Wouldn't that be the way forward? Or how do you see that? How do you see your collaboration with industry around this?
Yes, we already have some collaboration with some industries, and of course, we are not the only platform that do this. If you see, there are many startups and now more established companies that do have their own platform. I think that the COVID situation stress the system in a way to force an evolution. I mean, the role of the computational simulation in the entire drug discovery and development process is like this. Because the 90% is clinical, the 9% is preclinical, and then the design of the drug is the 0 point something effect. Now we see that it's possible not to revert, of course, but to integrate. So the level of the simulation, along with the quality of the data, so another fundamental point that I have addressed, just one point, is who is going to collect the data that allow us to design and model disease and stratify population. So it will be something that is in the end of the private. Government will take care of this and then decide in a way to have access to this data. So this is one of the major points. If we, the change here will be from try to model the data generated for other means and start generating data for the purpose of modeling disease and stratify. Because now we are reusing a lot of data that are not meant for the use of this. So we use just a small amount of this information. We have to change and decide if we want to move to a number to treat of 20% to a number to treat to 80%, we have to completely change the background. And the background is the profiling of the population, the understanding of the pathology. After 15 years, until 20 years, we do not know now which is the responding population for the rheumatoid arthritis. It's unbelievable. Yep. We do not know. We treat you with molecule 1. If you respond, it's fine. If it's not, we change. It is exactly the same that it was a century ago. And this is incredible. I mean, we go in the space with Tesla. Now we have Captain Kirk that arrive in the space, but we are not able to change the way we address pathologies.
So unfortunately, still some unanswered questions in the field of medicines. Unfortunately, I completely agree with you. Those massive diseases that are not all alike, but are really being treated as if they were alike. But there's definitely a personalized precision element in there we've not fully understood yet. And obviously, then the data becomes a challenge in that aspect. I think I'll just leave it for Jenny before I have a question in connection to that. Jenny, please, over to you.
Yeah, first of all, thank you so much for such interesting presentation. It's actually the first time for me to see the dynamic simulation in AR format in a demo section. It's very new. And since there is a belief that this new supercomputing technology can actually reduce the side effect for the tested participant in a clinical trial. Have you established any collaboration with the vaccination giants? For example, you mentioned there's actually investment in 90% in the clinical process. Have you ever combined this very well, I think is a very bright future technology in that process? Have some collaboration or communication with them, or your studies only based on the research outcomes?
I'm not in the condition to answer to your question now, but if you follow us in the next couple of months, you have some very striking.
Wow.
Because, as mentioned, we found something interesting in our simulation that regards the spike behavior that is, of course, related not only on the viral infection. And if he's confirmed anything, we have something to say, and is coming out from the simulation. So we are very proud to have, some success in addressing in a mechanistic way some of the side effects of the system. Also, in this case, the annotation is fundamental. So invest time in the annotate things allows you to see. So this information is already out there, but no one is collecting the data in the right way. So putting all these things together, then you see something, a pattern. And this is why we are trying to do this thing massively. Because when you have the complete set of the polypharmacology, you can see something. If you use Campbell data, you have the 0.0001% of the polypharmacology of the molecule. And you can see very local things. But with the limitation of the simulation, if you see the entire beast, you see something that is completely different.
Wow, it's very interesting. We're very looking forward to see the result. And so we won't let you enclose your business secret. Thank you so much.
And I have another, just 1 second phrase to say about the umbrella. In a meeting like this, I enter in the contact with the program manager of the umbrella program. And he said that it's just a chance that we have umbrella for rheumatoid arthritis, because this molecule dramatically fail in another indication, just because they miss completely the responding population. And in the case of rheumatoid arthritis, they have, by chance, the possibility to address almost only the responding population. So they arrive to a phase 2 clinical trial with a breakthrough, but for chance. If they arrive to the population that is not responding, now the most relevant molecule, disease modifying and world changer for patients, Enbrel, will never arrive to the patients. So this is something important to take in mind. So in that case, they are a 1 minute to fail because they are a startup at the moment in changing indication. If the chance they miss the responding population, now we do not Enbrel, and all the molecules are second, third, and fourth generation for that.
It's a very interesting concept you bring up here, Andrea, and obviously it talks into the value around real world data and real world evidence, if that is being accumulated in a sufficient quality. Because I think history in medicines has actually taught us over a number of years that a number of drugs have actually made it into the use by chance, almost by an unintended effect that wasn't the original, but it turned out to be the effect. So maybe there's a source of value out there in our clinical data. We really could access it, really could aggregate it, really could use it. So if I look across the European health data space and that ambition to build that massive access to data over the years to come, if I were to ask you, what would be your primary wish for that to support the journey you have for simulation and finding new targets for molecules? What kind of data? What would be on top of your list that you would like this to support you with?
For sure, starting from the profiling of the activity of the compounds. So we know very few activities. So when you put on the market a molecule is a PARP inhibitor, and you profile on kinase, you find that it's able to inhibit 100%, 200 kinase. So it's not a PARP inhibitor. No, it's a multifactorial molecule. And we don't know nothing about this mechanism.
Nope.
Think about cancer, when you derive resistance. Think that if you profile the molecule and you find that you have other 200 EGFR inhibitor in the nanomolar range. Now, already on the market, already can be used, that overcome the mutation that you see in that patient, which is the difference here. So just limiting on the profiling of the molecule. If you invest as a community in the full profile of safe in man compounds, and then we generate 300, 400, 10,000 data, high quality, dose response data. What is the outcome for the patients? It is huge. I mean, then you can repurpose molecule by the meaning of the data. You can combine molecules. You can predict side effect, because if you have the polypharmacology, you can predict side effect, drug-drug interactions. Anything is there. So it is an investment that is limited. We are not talking about billions. We are talking of several millions respect of the cost of the sanity. So it's really a drop in the ocean. But what is the change in the paradigm shift in seeing a molecule in patients if you have access to these high quality data? And also the variability. We don't have these molecules. So in COVID, we test the 30% of the molecules that are variable from the history of pharmaceutical. We have to collect these compounds to be used for neglected, infected disease, population, developing country. So, raloxifene costs a fraction of any novel molecule identified. So the treatment is less than $30 per treatment, in case of success, of course. But it's one example. So also in this case, now, how many people we can treat if we can reuse molecules that are expired for other indications?
No, that's definitely interesting from a safety perspective of tolerability and toxicity, and no molecule well understood from that profile, but may actually turn out to be beneficial in new circumstances. Fully appreciate the potential that is in that, and also the value it proposes to the healthcare systems across Europe and for the patients equally alike, and also for us as regulators. It actually makes it somewhat more substantial in the evidence, using real-world evidence in that aspect that way around as well. Interesting journey. I think we're up on time here. I really appreciate your presentation here today, and obviously you gave us a cliffhanger for the next week and months to watch out for. So really looking forward to that. Thank you so much for joining here today and presenting your initiative.
Thank you very much for the invitation. Very nice discussion. Thank you very much.
Thank you. Bye-bye.
Bye.